We've been researching herpes simplex virus type 1 for probably 16, 17 years. And I've been working on bovine herpes virus 1 for about 25 years. About 15, 16 years ago, we discovered that the genes that are expressed during latency by bovine herpes virus 1 interfere with the virus killing neurons. We then looked for similar function in genes that are expressed in latently infected neurons of HSV1, and we found that HSV1 genes expressed abundantly in latently infected neurons also encode a product that interfere with programmed cell death. This is important because both viruses, when they establish a latent infection, they need to keep the neuron alive for latency for two reasons. One, the virus has to stay there. And two, neurons are terminally differentiated cells and they can never divide. So if the virus kills that cell, then it no longer has a place to have a long-term latent infection. We still continue to try and understand how this influences a long-term latent infection and how we can utilize this to make vaccines, especially against bovine herpes virus, that protect animals from infection but prevent latency from occurring. Most of our efforts are working on an animal herpes virus. A lot of things that we find in that animal herpes virus are directly related to how the human herpes viruses behave. It's become probably a bigger problem than it was 25, 30 years ago because people are using modified live vaccines of bovine herpes. And these modified live vaccines, although they're effective in preventing disease in adults, it's also clear that they are responsible for increasing the incidence of abortions in pregnant cows. And that continues to be a real problem.
Dr. Jean Defay agrees that vaccine-induced abortions are a problem. He has been researching bovine herpes virus, looking at the genetics behind the disease.
I think it's fascinating to sort of try and understand what the genetic basis for disease. We're looking at the genetics between them and finding out what is it that might be responsible for the pathogenicity, for the increased virulence of some viruses. Starting a number of years ago, veterinarians around the country realized that the vaccine that's available commercially for IBR, for bovine herpes virus 1, under certain conditions could have caused abortions in cattle. So we started trying to understand whether we could identify, separate or make a difference between the vaccine strain that was given to cattle and the wild type, which is the virus that circulates throughout and causes disease. I think that by looking at genes from viruses that cause severe disease and those that don't, and look at the genes that are present in vaccines, we may get a better understanding of what is it that determines the ability of a virus to cause severe disease, and what can we do to make the virus attenuated to use as a good vaccine that's not going to cause abortions and that not going to predispose them to respiratory disease in feedlots.